Today, I'm very excited to talk with Professor Cynthia Moss. She's at Johns Hopkins University, where she's in the Department of Psychological and Brain Sciences. And with her is Daniel Kish. He's also a scientist. Daniel, I did a little reading up on you. He got a master's degree in psychology. And he's doing a lot of work now on human echolocation. Dr. Moss, Cynthia, is an expert on bat echolocation. Of course, bats, they can't see too well. You know, there's this, I remember growing up, maybe my mom said this a few times, blind as a bat, right? But they can navigate extremely well in three-dimensional environments, even though they can't see well. And they do that by echolocation, emitting sounds and then having those sounds bounce off of something and come back. So Cynthia's, I guess she's, you know, she has kind of a background like mine in a way. My bachelor's degree was in zoology. My PhD was, I studied a neuroendocrine system that controls molting in crabs. And then I did some work on snail brains before moving to mammals. And so this kind of maybe comparative neurobiology background, I think, is very valuable for kind of understanding the whole field of neuroscience. So that's what we're going to talk about today, going from bats to humans and echolocation. Daniel, so he went to college at UC Riverside. When he was young, he had, I guess, retinoblastoma. Is that what it was? It's essentially cancer that selectively affects the eye. So he's been blind from a young age, but he's developed the remarkable ability to do echolocation. And he's actually, what's the organization? He's president of the World Access for the Blind. Is that right? So
Yes.
You founded that effort, yes. And it's an effort. And he's, you know, my understanding is he's involved in helping people who are blind learn to echolocate. And it turns out they can do it pretty well, and we'll hear about that from Danny. Okay, yeah, so go ahead.
Well, a couple things. So yes, it is true that we help blind people to echolocate. What we're really about is self-determination and personal freedom, of which echolocation is a component or subset, because it's certainly a proficient echolocator has the opportunity to gain more freedom of movement and also more security. And I'll get into that a little bit more. I'll clarify a couple things. My second master's is in special education. So as a development psychologist, I educate, right? So I kind of combine both disciplines with a sprinkling of other disciplines to help people learn. I think of myself less as an educator in a way and more a facilitator of learning. I would, though, like to address this blind as a bat thing.
Yeah.
And Cynthia knows a lot more about it than I do, so I'm gonna punt it over to her. But it's a bit of a misnomer. It's a bit of a misconception. Yeah. And I actually made that mistake years and years ago. One of my first presentations back in 2000, and I kind of made the thing blind as a bat, and that's how I started my presentation. And it was a 12-year-old who piped up and said, "Yeah, bats can actually see pretty well." And I thought, okay.
Yeah, so I can elaborate. There are about 1,400 species of bats, comprising nearly almost 25% of all mammalian species. And about 200 of those bat species do not echolocate at all, and they rely on vision. And of those that echolocate, many have reasonably well-developed vision that they use probably for navigating long distances, recognizing landmarks that could be tree lines or mountains. And
is that even in dark, when it's dark outside, they still use their vision?
they can, it's rarely pitch black. There's moonlight, starlight, artificial light that gives bats visual information.
They tend to favor dusk, right? When they're out hunting, they tend to favor dusk.
Well, that's when we see them.
Oh, okay, fair enough. So we see them. Okay.
Yeah,
okay,
but they are out during, like in Scandinavia, where there's very little darkness during the summer. Right. They're flying around it in the light. Yeah. And I'd say the bats that rely heavily on echolocation, their vision is, for the most part, comparable to rodents. And many researchers use rodents as subjects for vision research. So, so they're not so visually disabled, but they have complementary vision and echolocation for pursuing food and steering around obstacles and so on.
What's remarkable is how complementary the senses work together in bats. With humans, by comparison, are highly visual, and we use our hearing for very specific kinds of tasks. But vision is often regarded as humans' primary sense for spatial awareness, primary sense for getting around, primary sense for accessing information. Whereas a bat, lots of species of bats just really share those functions across sensory modalities. And in a way, that's really the remarkable thing about bats, is that many species of bats have developed their hearing to such a degree that they're almost happy to, use one or the other. If you put a bat in pitch blackness and they're an echolocating species of bat, they'll echolocate.
Yeah.
And certainly echolocation provides more useful information for finding insects in the dark. The visual information is going to be rather minimal. But for navigation in particular, I think vision is important to many bat species.
Yeah.
So you said there's like 1,000 or more species of bats, and so I looked on the internet trying to find, their evolutionary history, and it appears it's not completely clear. They're the only flying mammal, right?
They are, with powered flight. They're gliding mammals, but bats are the only mammals that have powered flight.
Probably evolved from some non-flying mammal, I would assume, but it's not completely clear. Is that right?
Right. And, because the name bat in many languages includes the word mouse, in German and French and in Swedish, many people think of them as flying mice, but they're not related to mice. That's
another misnomer.
Yeah. Okay. And what are some of the I guess, structural? So bats tend to have big ears, right, relative to their size of their head, and relatively small eyes. And what other kinds of adaptations do they have for echolocation?
Well, the auditory pathway is a mammalian auditory pathway, but there are specialized circuits that operate to extract information from echo. So for example, bats are they rely on the time delay of returning echoes to measure how far away an object is. And they show extraordinary ability to discriminate between echoes arriving at different delays. So they can discriminate differences in the microsecond, maybe even the sub-microsecond range. It depends on the experiment. So presumably there are specialized circuits for extracting that temporal information. And they have a lot of brain tissue devoted to processing sound. But they have the same overall brain organization as humans.
And they emit so we can hear
ultrasonic.
Sorry,
right? So they emit high frequencies, and they can hear those high frequencies, whereas humans, we don't hear into the ultrasonic spectrum. And we don't have, I mean, even proficient echolocators, as far as we know, don't have that specialized brain circuitry that bats have. What we do have, or so it would seem, is the capacity to interpret the heck out of data that come in, to extract, to kind of interpolate enough information to form an image, to construct an image. So while the overall mammalian brain pathway may be comparable, the specific circuitry, God, bats have had, tens of millions of years head start on humans in developing that kind of largely auditory circuitry. But humans were pretty adaptable, and we have pretty complex brains, and we've found ways of compensating for that pretty well, I think.
And Daniel, so did you start learning to echolocate soon after you went blind, when you were young?
Yeah, well, by my parents' reports, I lost my first eye at 7 months and second at 13 months. My vision would have been very, very damaged, though, prior to the enucleation of the eyes. So I was probably, as an infant, shifting perceptually from the small amounts of vision I had to becoming non-visual, especially as my vision deteriorated very, very rapidly. I have no memory of this. I'm kind of guessing at this. By the time I was 15 to 18 months old, my parents say I was clicking, and that I seemed to evidence an awareness of what was around me. It was easy to do because I was an early walker. So I was walking by 7 or 8 months as an infant. And my earliest memories go back as far as about 2 or 2.5, when I distinctly remember echolocating.
And so Daniel started producing sounds and discovered that they returned useful information from the environment. And we just yesterday had a discussion with members of my lab, and Daniel referred to learning to see. And a lot of I could see the questions on the faces of people in my lab. What, learning to see? You don't have to learn, it just happens. But, I think there is this developmental process where you learn to take in sensory information and use it to guide behavior. Yeah. And we take this visual learning for granted as visual mammals. Mm-hmm. And I think something similar operated for Daniel with the use of sound. He learned to use sound to get around.
That's amazing. And so the brain, when you're really young like that, the brain is more plastic. It's better able to kind of, in this case, start using and actually forming, modifying neural circuits that previously wouldn't have, maybe be used for vision, right? Because you can't see. So those circuits probably somehow are playing a role in this echolocation, that your ability. But the other point on that is, so in your own work so you're working with children a lot, because it seems I would think the earlier they start to learn to echolocate, the better they'll be at it.
Well, again, it's about self-determination. I mean, it's a much bigger pie than just echolocation. The whole process of one's own agency over one's own movements, over one's own choices, over carrying out choices, taking action on the environment. That's a thing that sighted kids take for granted, that blind kids often have to really fight for. And it's often withheld from blind children in many, many different ways. Yeah. Echolocation is one way of kind of reclaiming one's sense of self-determination, one's sense of personal agency. The saying I like to say is, the earlier, the easier, but it's never too late. So
Yeah.
The reason I work so much with children, I am a child specialist. I mean, that's one thing. But it frankly has more to do with children are most often referred to us and they're the easiest to get funded. We do work with older people. A good 30%, give or take, of our clientele are older people. My personal oldest client was 83.
Wow.
He did great, actually. So and as far as the brain stuff goes, I mean, probably Dr. Laura Thaler has done, I'd say, the most up to this point anyway on the brain scan research and the neurological underpinnings of echolocation. And yes, absolutely, there's very much within the visual system that is supporting the echolocation process. The reality, though, is that it's a whole brain function. Because the brain has to do all kinds of things. It has to interpret, it has to recognize and establish context. It has to determine what the relationships are between, self and object. It has to prepare for some kind of motor function to occur. And then strategize how that's going to occur. So there's probably a lot of higher brain function involved in a blind person just figuring out how they're going to go get a glass of water, So, but absolutely, the visual system is quite keenly involved in this process. I call it the imaging system. So I think of the visual cortex as an imaging cortex, and I often refer to it as an imaging system because I don't think it's confined to the visual cortex. We know it's not confined to the visual cortex. So there's a whole system of brain engines, let's call them, that are responsible for this kind of imaging, and then acting upon governing action upon those images.
Yeah, and so we'll get in. I think we'll wait a little bit before Cynthia starts talking about her electrophysiology recording from freely flying bats. But Cynthia, why don't you talk about your lab? What's it look like, and how do you study echolocation? Can you are these bats friendly if you're around them all the time, and kind of what's the setup and how do you work with them?
Yeah, so we actually have 3 different species of bats in the lab right now. Egyptian fruit bats, which are highly visual, and they make echolocation tongue clicks instead of vocalizations like most species. So tongue clicks similar to Daniel and other echolocating human signals. And then we have big brown bats. And the big brown bats are found throughout North America. They're insect-eating bats. They produce echolocation calls with the larynx and transmit them through the open mouth. And these bats, the name is a little bit misleading because they're not big. They weigh maybe under 20 grams for the most part. So they're about the size of mice.
Yeah.
And then we have another echolocating bat that's from the neotropics that produces echolocation calls that transmits them through the nostrils and has a little nose leaf that allows it to sort of move the directional aim of the sound beam by moving this nose leaf. They feed on fruit, and they probably have a little bit better vision than the big brown bat, but the eyes of both species are rather small, whereas the Egyptian fruit bats have quite large eyes. And so,
yeah, they don't. The fruit tends to stay in one place.
Yes, exactly. But they probably use echolocation to steer around obstacles. They may be foraging in dense vegetation, and so they'll certainly use echolocation to get around, along with vision. And of course, they need to determine which fruits are ripe and ready to eat. So they'll use olfaction for. In my lab, we have a large room. It's about 6 by 6 meters that's lined with acoustic foam on the walls and ceilings, and then it's carpeted. And in this room, we have lots and lots of high-speed cameras and microphones so that we can quantify the animal's flight path, maybe in a task steering around obstacles, it may be going after some food, depends on the research question and the species we're using. We have, with the microphones, we can collect data that allow us to reconstruct the sound beam pattern for each call, so we can see where the sound is directed. And we find that bats actively control the direction of the sound beam, just the way we control sort of the direction of our visual gaze, where we point our eyes. And so they use the control of the sonar beam to inspect objects in different locations. So that's just sort of a general overview of the lab. We have another set.
And do you have are you able to have like a moving target, or you have food that's sitting in one place? And
we've done both. We've run some experiments with free-flying insects, and we've attached tethered insects to systems that produce some movement. We're actually in the process of setting up a trajectory control system so we can have a tethered insect that moves in a path that we define before the experiment, to look at the bat's flight path and whether it's showing some planning for interception in flight. And we have another setup that is in a smaller room, again, with lots of high-speed cameras and microphones. And in this setup, we currently train bats to just perch on a platform. And we use this exclusively with the big brown bats that seem to be comfortable just sort of sitting and waiting for food to arrive. So they can track a tethered target that's moving along a path. And we look at the changes in the echolocation behavior as it tracks the target at different distances. So one of the things we observe both in the bats that are perched and flying is that they change the features of their calls as the distance between the bat and the target changes. So as the target distance decreases, the call rate increases and the call duration decreases. And so we see this pattern in both the perched bats and the free-flying bats.
And then you've been able to, and this I'm really interested in, so you're able to put electrodes in their brains. I think you've studied mainly two regions, one, which we haven't mentioned yet, the hippocampus, which is very important in spatial learning and memory. And actually, I think I was going to ask Daniel about this. So you must have some sort of you obviously have some representation of environments you move through. You move through an environment, you echolocate, you remember what's where, right? And then you kind of, is it like visualizing it? Same kind of
it is. If you've ever seen some of the demonstrations I've done where I move through an environment that I haven't been to before, and I echolocate that environment and describe it. So I basically do a scene analysis of the environment, and then I draw it or sketch it. And the sketches are always from a bird's eye view, really. Yeah. So all the relationships between objects and certain characteristics of the objects. I mean, I'm no sketch artist by any stretch, but I'm told that when you look at the sketches, they do in fact represent the environment. So that's a way of giving people a window into into a non-visual imaging system.
I had Cynthia, I had Jim Knierim on one of these, talking about place cells and grid cells and that. And you've been able recently, I see, to record from the hippocampus of freely flying bats. Do you have like Bluetooth, or how do you get the signal coming from the? You don't have them on a cable because it's going to get all messed up.
No, we don't do. Yeah. So we have a commercial telemetry device that we've used to record from both the hippocampus and the superior colliculus. The superior colliculus is a brain structure that's commonly thought of as implicated in ocular motor control, but it's actually more appropriately considered an orienting structure. And so this different animals have different orienting behavior. So for a visual animal, the movement of the eyes and processing of spatial visual information is going to be mediated through the superior colliculus. In the case of the bat, we find that there are neurons that encode the 3D spatial location of objects, and premotor activity is tied to the production of echolocation calls, head movements, and ear movements. But yes, many years ago, we actually did some experiments recording from the hippocampus of a bat that was crawling. And these experiments were done in animals that were tethered, but they could crawl around an arena. And this work was done by my postdoc, Nachum Ulanovsky, who really piloted the first hippocampal recordings in the freely moving bat. And he was very careful to try to replicate all of the experimental methods that were used at the time with freely moving rodents, just to be able to compare the species directly. And he did find place cells in the crawling bat. And he also discovered that the place cell tuning sort of changed over time following each echolocation call. So immediately after the echolocation call, the tuning was sharper, and then over time, that tuning sort of broadened. He went on in his lab to do the first recordings from the hippocampus of the free-flying Egyptian fruit bat. So this is a much larger species and easier to get recordings from animals because they can carry heavier devices. And in his lab, he's used telemetry to some extent, but also stored the neural recording data on the animal and then downloaded it after the experiment. And so we've used this a telemetry device to record neural activity from the hippocampus and the superior colliculus of the free-flying bat. They can carry 3 to 4 grams in flight, but it is a large percentage of their weight. So Nachum refers to his shift to using the Egyptian fruit bats that weigh about 150 grams as the low-tech solution to the challenges of recording from a flying bat. But the data we've collected from bats, in many ways, from the hippocampus are comparable to what has been reported in rodents. But in the bat, we see place cells that show, 3D response areas.
Okay. And so these bats are emitting these high frequency vocalizations, so in a frequency range mostly that humans can't hear. And so can you talk a little bit about kind of the physics of this? So you have frequency modulation, so they can change the frequency of the emissions, I guess, so they can change the length of time of a vocalization, how long it lasts. And there, so can you talk a little bit more about the physics? Because it's
Sure. Yeah. So I mentioned that there are over 1,000 species of echolocating bats, and they use a variety of signals for echolocation. The bats in my lab, the big brown bat and the Seba short-tail fruit bat, both produce frequency modulated signals. They sweep in frequency from high to low, and in a very short period of time, just in like a few milliseconds. Wow. There are some bats that produce constant frequency sounds that are more like long tones at a set frequency. And then these constant frequency components are flanked by frequency modulated components. But let's just for now focus on the bats that use frequency modulated signals. We refer to these as FM signals. And in the big brown bat, as it's searching for prey, it produces calls that might be 5 to 10, maybe out in the open field, up to 15 milliseconds in duration. Once they detect an insect, they immediately shorten the signals. So and I should add that the longer signals tend to have a shallower frequency modulation. They may sweep from 30 kilohertz to 25 kilohertz in 10 milliseconds. Once the bat detects an object, it'll increase the overall bandwidth, the start frequency of the fundamental. And these are frequency modulated signals that have multiple harmonics. The fundamental will sweep from maybe 60 kilohertz down to 25 kilohertz in just a few milliseconds. And then as the bat prepares to take the insect, the calls are produced. They're maybe half a millisecond long, and they can drop in frequency a bit. So now the highest frequency in the fundamental may be 30 kilohertz, 35 kilohertz, and then sweep down actually into the audible range. So humans who have very good high frequency hearing can actually pick up these signals during that final capture phase, which is often referred to as the terminal buzz. So there are a few reasons why bats adjust the duration and the rate of their calls as they approach an insect. One, they tend to avoid overlap between the outgoing call and the returning echo. So if they're far away from an object, they can produce a longer call and avoid that overlap. But as they get closer, they need to shorten the call to avoid overlap. They also wait for the echolocation, the echo to return from their call before producing the next call. So when they're far away, they have to wait longer for the echo to return. Once they get closer and closer, they have shorter and shorter wait times. So they're continuously modulating both the duration and the rate at which they're calling with respect to the distance to the object that they are inspecting and perhaps preparing to intercept, if it's an insect-eating bat that is preparing to capture its prey.
And then Daniel, how do your vocalizations compare to the bats?
There are a lot of similarities. Obviously, humans don't tend to use frequency modulated signals or chirped signals. Humans tend to use clicks because that's just what we can orally produce. But those clicks are complex. So they contain a composite of frequencies that have a lot of advantages, it is believed, in terms of gathering different types of information from surfaces. They are also very short in duration, not as short as the bats, but tongue clicks such as I just produced, which is a palatal click, has been measured at around 5 milliseconds, with peak frequencies in the areas between kind of 6 and 10 or 6 and 12 kilohertz, with some frequencies down into the lower range as well. But there's a very, very, very sharp attack, very sharp onset. And that sharp onset mathematically produces a very wide spread of frequencies. Okay, that tends to be consistent. The timbre, the spectral content of the signal tends to be consistent for many echolocators across activities. What changes, though, is often the amplitude of the signal and the repetition rate of the signal and the directionality of the signal. So humans will scan a lot with their heads. Bats will also scan with their beam, not necessarily so much by head movement, but by trajectory of flight, and also by their motile ears, shaping their ears to capture sound. If humans could shape our ears, we would, but we can't. So there's a lot more head movement involved. And then the amplitude variation is often used for changes in environment, such as noise. So if you're in a noisy environment, you increase the amplitude to essentially cut through the noise. So you're imaging through the noise by, forcing sound, if you will, through a noisy environment, but also for objects at long ranges, as same with the bats. So if you're looking for objects at further ranges away, you need more acoustic energy to reach those surfaces. It's one of the reasons why bats use longer chirps as well. They don't need the echo, they don't need the signal to be out of the way of the echo, but arguably they can also emit more acoustic energy in that pulse. And humans just increase the amplitude of the click, right?
And I can add that bats in noisy environments also increase the level of their signal so that they make these adjustments. And some species, the big brown bat being one of them, do move their heads to scan.
Right.
Yeah.
Okay. Yeah. Right. And then the other thing, though, is with echolocation, we often think of it as detection. You detect surfaces, you detect features, you detect items. It's really not detection, it's discrimination. You're distinguishing or discriminating a feature from its background, or an item from another item. And so as I often refer to it as the difference between targeting sonar and orienting sonar. So when you're targeting something, you will often produce softer sounds because you want to, humans at least will, because you want to decrease the amount of background and increase the feature, the foreground item. And a way of doing that is to decrease the amplitude of the sound so that you can kind of focus on the feature of interest. And humans will often increase their click rate as well. So now they're scanning, now they're focusing. It's edge detection, it's feature detection, it's contour detection. And those things require more scanning and faster repetition rate. Another reason for more rapid repetition rate would be if you're moving quickly, or if you're moving in a dynamic environment where a lot of people or other objects around you are moving quickly, then you need faster spatial updating. And again, you're looking at more scanning and you're looking at higher repetition rates.
Yeah, so there are a lot of similarities there. The biggest difference being the frequency content of the signals used by bats that are hunting very small insects. So they require high frequency sounds with very short wavelengths to return audible echoes. Humans are listening for echoes from larger surfaces that they can pick up with the lower frequency sounds.
Right. So the spatial resolution of the bat system is quite a bit better than humans.
Sure, for sure. Yeah.
But over time, Daniel, have you been able to increase your spatial resolution? I'm going to put in the description section links to videos from Cynthia's dad and Daniel. She had you in her lab. I saw one video where you have, I don't know what they are, PVC tubes or something.
Pipes. Yeah, the pipes.
The pipes. And so with increasing practice, do you get better? I mean, that's...
You do get better. You do get better with practice. I mean, I'm not hunting insects yet. But you know, I mean, even as a kid, you know, I remember chain link fences, for example, wire fences being quite difficult to detect. I could run into a wire fence. So as a kid, I was mostly looking for the posts that held up the fence. And I'm sure I had excellent hearing as a kid. I mean, why wouldn't I have excellent hearing as a kid, a 10-year-old? As an adult, I can detect those fences. So I think a lot of it has been, as a mobility instructor, I've had to just really step up to the plate in terms of what I perceive, how I perceive, the subtlety, the nuances of sound that I've learned to perceive. Yeah, I often call it the first axiom of neuroscience: use it or lose it, but the more you use it, the better it gets.
Yeah.
And even as I'll be 57 in a couple of weeks here. I dare say that my hearing is as good as it was when I was a kid, but my functional hearing is better than it was when I was a kid.
That's amazing. So I can't remember when this was. It was a few years ago. My daughter had this thing on the internet where you can test how high frequency sounds you can hear. And, I'm definitely losing my high frequency sound perception, but and your clicks, actually, I couldn't. What frequency do you know?
Well, I mean, Zoom may not, capture these clicks very well.
But maybe try getting a little closer to the computer.
Well, how about this?
Yeah.
So that's a lower frequency click that many echolocators do use. It's called an alveolar click. And it has many of the same characteristics. It's quite rapid, has a very sharp onset. It's just lower in a frequency. The spectral content is lower in frequency. Can you hear that?
I can't hit. Boy, I got to get my hearing checked.
No. My guess is that Zoom is compressing.
Okay.
It's just not a good medium for these kinds of transmissions.
Okay.
But Daniel does have a very crisp, intense click.
Yeah.
I can't. I guess I could learn, but I haven't yet learned how to make such crisp, intense clicks. I think that's one of the things that Daniel instructs people on is the click, because the click determines the information that returns.
Yeah. I often say the more clearly you interrogate your environment, the clearer will be your answers. So echolocation is about querying or interrogating your environment for information. The more clearly you do that, the more clearly the clearer the calls are, the clearer is the information that you get back.
Now in the bats, they don't just make vocalizations for echolocation, right? Do they communicate with each other?
Absolutely. And of course, the social calls are quite different across species. One of the things quite interesting about the Egyptian fruit bat is it uses tongue clicks for echolocation and vocalizations for social communication. So that's very easy to tell what's what. In the big brown bat, both the social communication sounds and the echolocation sounds are produced with the larynx. And some of the echolocation sounds and social communication sounds, just looking at spectrographic displays, look quite similar, but they serve different functions. The bats can hear the differences. And if we slow the sounds down, we can actually hear the differences between the social calls and the echolocation calls, even though they're both perhaps frequency modulated calls.
Are there any bat species that hunt predators that are fairly large, and do they cooperate with each, like wolves or something like that to?
No, I think so there are many different diets of all the different bat species. And there's some evidence that bats that feed on fruit may forage cooperatively. They may fly together to a site where there's a lot of nice ripe fruit. There are some bats that feed on frogs and maybe lizards. There's some bats that take fish from the surface of water. There are vampire bats that they take blood from mostly farm animals. And of course, then they
get fish. So do they use echolocation to get fish? How does this
so these are, I think what they're doing is they're listening for echoes that carry information about the ripples in the water surface that are produced by the fish.
Wow, that's intriguing.
Yeah.
No, I guess what I was thinking is, so there's some, like dolphins use echolocation, right? And do they work together to, like, I don't know what you call it, corner or move schools of fish to?
Yeah, I think there is some anecdotal, or maybe more than anecdotal, there's some evidence that dolphins do that. In the case of insectivorous bats, I think, each bat for him or herself. They're not really cooperating,
Yeah.
but they may find that there are lots of insects in a particular area, and there'll be many different animals foraging in that area. But they tend to distribute themselves so that they're not interfering with each other so much. Although some years ago, I did witness some insectivorous bats in Belize that were foraging in real close proximity around a street lamp where there was a lot of insects collecting. And that maybe, I mentioned that the bats produce what's referred to as a feeding buzz as they're preparing to capture insects. And there's some evidence that bats will listen to the feeding buzzes of other bats to know where the food is.
Right.
So, so they may attract other bats, but I wouldn't say that they're cooperating, but they may listen in and get information that's useful for discovering a good foraging site.
So you mentioned the insects kind of at nighttime accumulating around light. They're attracted to the light, right? And I've noticed this over, I don't know, the last decade or so, that you go out in summer at night and it doesn't seem like there's as many insects around the lights. I've seen, there's a lot of studies emerging that there's a decline in a lot of species in general, but maybe insects in particular. Is there any do you know what kind of work's going on with bats and how the difference? I heard about this fungus affecting some bats and
Right.
so.
Yes. So the white nose syndrome, a fungus, has wiped out millions of bats in North America. And there has been a lot of research on its fungus and its impact on bats. I don't know the latest, but it seems maybe over time, the bats that do survive have some resistance to the fungus. And in Europe, I believe there are funguses similar to what's killing bats here in North America that doesn't kill the bats in Europe, perhaps because some period earlier in time, they evolved some resistance to the fungus.
That's interesting.
And bats we hear a lot about bats with viruses, right? Latest, the SARS-CoV-2, but MERS, Ebola. So they have something about, I don't know, their immune system that lets them harbor these viruses and not wipe out the bats, but apparently that applies to viruses, but not fungus. I don't know.
Yeah, I guess so. But bats are remarkable animals in many ways. We talked about how long they live. There's generally a trend for the lifespan to be related to the size of an animal. So if bat is the size of a mouse will live roughly 2 years, and a bat can live 40 years. It's amazing. And then, as you say, they can harbor viruses without dying. So their immune systems, there's a lot we can learn from their immune systems. And
don't their wings also heal?
Yes.
With extreme speed, the membranes on their wings?
Yeah. So they do, they can even fly with some holes in their wings, but they do repair. And then we were just talking about human hearing and how it's common for humans to lose high frequency hearing with age. It seems that bats have some resistance to hearing loss, because of course it's so important to their survival.
Yeah, I mentioned this before we started the podcast, but I think I'll bring it up again. There's this interesting story in relation between lifespan and uric acid in the blood. And people may have heard of a condition called gout, where this chemical, uric acid, gets really high levels and essentially precipitates out and forms crystals that accumulate and cause inflammation around joints. But it turns out that in bats, birds, and somewhere in late primate human evolution, humans, there was a mutation in gene encoding an enzyme called uricase, which gets rid of uric acid. So mice have very low levels of uric acid because they have uricase, gets rid of it. They have short lifespan. Bats with their equivalent body size, as you said, they live 40 years instead of 2 or 3. And then, of course, humans live longer than others. And it turns out uric acid is the most prominent antioxidant chemical in our blood. It scavenges free radicals, hydroxyl radicals. And there's actually even some evidence correlating uric acid among humans with intelligence. Now, it's actually pretty good data, but and people with gout actually tend to live longer. So this is all. So I came to your lab
very uncomfortably.
It was 20 years ago. I came to your lab, and I actually had one of the people in my lab get rabies shots. We're thinking maybe we'll end up collaborating with you, but we've done some genetic manipulations of uricase in mice and found effects on lifespan and aging. But I think the genetics, I think this is a really important area, right? The genetics of bats and being able to manipulate genes and see how they affect, whatever, aging or echolocation or whatever. Are there labs that are trying to do that kind of thing? In other words, make a knock out this gene and knock out uricase in bats.
Yeah, so I'm not aware of any knockout studies yet in bats, but there is an intense effort to really map the genome of different bat species, and that could lead to this kind of work down the road.
Yeah, I had oh my gosh, I'm really bad on names. I had an investigator at Rockefeller University who studies songbirds, right? And I guess they're at a stage now where the people who work on the birds have gotten to where they can start to manipulate genes in the birds. Actually, CRISPR, that may be a way to avoid having to go, like the germline manipulations of knocking out genes, because you can do things in actually even in adults, in theory, manipulate the genes. Do you know if, we're getting really into the science here, but so we use a lot adeno-associated virus as a vector to either introduce genes or put CRISPR constructs so we can knock out a gene or even correct a genetic mutation. And the AAV selectively infects neurons, but it doesn't hurt the neurons. And so I think that would be the way to go in the bats. You just inject the viral construct into the whatever neurons you want to.
Have done some of that for doing things like 2-photon imaging and dreads. So trying to manipulate, reversibly inactivate brain regions or monitor activity in brain regions. So we're starting to do this work. So some of the genetic tools that were developed initially for mice and were sort of used to sort of justify the focus on mice as the neuroscience research model now really can be used in a variety of species.
Yeah. Of course, always in science, a big thing is funding, right? And, I guess anytime you can kind of keep up or advance the technology, like you develop ways to record neuronal activity from individual neurons in freely flying bats. And that's a big advance that allows you to really get into what's going on actually in the neural circuits. And then maybe a next step would be manipulating some gene, like uricase. I'm retired now. Otherwise, I'd write a grant with. Or I wouldn't have to actually. I was a lab chief at NIH for 20 years, and now I'm just an adjunct professor at Hopkins. But so I don't have a lab. Let's see. Oh yeah, there was one last thing. So what can people who are interested in, I guess, what would be the military sonar or so I guess you'd say artificial echolocation technology. Is there work going on in that?
I think there've been
Well,
all kinds of attempts.
Yeah,
in
there's
that
certainly
direction.
lots of work. Much of it is classified, so I'm not privy to the details. But one of the things that, you know, we talked about earlier, the changes in echolocation signals with distance or with the task at hand. I think that's key to the success of animal echolocation, including human echolocation. And to the best of my knowledge, that is not widely implemented in the Navy sonar. They tend to use their fixed signal again and again and again. But there may be behind the scenes efforts to introduce adaptive sonar signal design. And that may, I think that holds a lot of potential for applications in monitoring the underwater environment. And sorry, I've got a frog in my throat. So we'll see.
If there's a wall, so so I'm in my house now, and behind this wall is another room. And so is it possible to emit a sound and have it go through something, hit something that's behind, say, a wall?
There was an ultrasonic device that was designed for humans, and it produced signals quite similar actually to bat signals. They had this demonstration, they called it the Pepsi Challenge, and dolphins can do this too. But the idea was, is the can empty or full, or half full? And all you did was you aimed the beam at the two cans, and you could tell very clearly how much fluid was in each can. And I think there's evidence that suggests that dolphins can do a similar thing.
Yeah, I think it depends on the material.
Yeah.
So sound can travel through some materials and not others.
Right.
Yeah. Okay. I think we've covered a lot of ground, and I'm going to put links to videos and websites for both of you, so that people can get more information and see some of your lectures, Cynthia.
Okay, great.
And videos of Daniel's work. And yeah.
And will you be sort of editing what we've talked about to create the podcast, or is it just going to be straight recording?
No, unedited.
Unedited. Yeah. Okay, very good.
Yeah, I intentionally did that from the beginning. That's the idea. Is this is a collection in one place of conversations between two neuroscientists. Mm-hmm. One who's trying to learn about something in neuroscience that he doesn't know much about, me, and the other person who can teach. And then, so these are really targeted a lot to, I'd say, undergraduate and above with an interest in neuroscience, brain science. Mm-hmm. And so I get big shots like you, Cynthia and Daniel, and have a collection all in one place.
Well, very cool. So please do send us the link once it's out.
It'll be soon, probably tomorrow.
Oh, okay, great.
Have a great rest of the weekend in Baltimore, Daniel.
Thank you.
Yeah.
Thanks very much. Yeah, we've got to get back to this design challenge that we're involved in. There are undergraduates and graduate students who are participating in a design challenge in assistive technology for visually impaired.
Right.
So we've just
Interesting.
ducked out for a few minutes to talk with you, and now we'll go back to see.
Well, thanks for taking the time out, away from that, and yeah, go get back to it.
Yeah,
Okay.
yeah, really nice to talk with you. Thank you.
Thank you.
All right, bye.
Bye.